So I was asked to talk about how we can start to chart evolution of tumors throughout the therapy using novel means, including and particularly application of genomics to the use of circulating free DNA. So what we have discovered over the years is that colorectal cancer, metastatic colorectal cancer, is a complex disease involving different subtypes. And some of these subset of tumors have hallmarks that can be drugged. For example, BRAF mutant colorectal cancer, HER2 amplified colorectal cancer, or colorectal cancer that have translocation in specific genes. And these oncogenic addiction can be useful in the clinic because you can go after them. So that's the main topic. And what we discovered over the years is that although you get responses, profound responses if you get the right oncogene in the right person, then you will get relapses and eventually the tumor will come back. And so it's really what all my work is currently ongoing is focused on understanding how tumor evolve during therapy and how we can start to chart this molecular evolution and how we can start to go after molecular evolution. And this brings us back to understanding genomic diversity, understanding instability of the tumors, and understanding how the tumor can survive a therapy, which is really why tumors kill. As you know, I'm not a clinician, but I interact with a lot of clinical fellows. And it has been incredibly rewarding for me to be able to translate some of the findings that we made into the clinic. These include, the use of BRAF or NRAS or KRAS mutations as diagnostic tests. Moving into the application of new methods to understand and discover resistance before it gets manifest. So sometimes we can now see, before a CT scan can detect it, the relapse of the patient in the blood of the patient. And we think this may help in the future. The next step is to understand, profoundly understand and believe that what we should be after are not oncogenes, but the ability of the tumor to evolve. If we understand that, then we start to have new ideas on how we can stop tumors. And that will involve certainly finding the right oncogene and finding the right drug for that, but also unleashing other means to stop the evolution. We need to block the ability of the tumor to escape therapy. That can be done by using mutations that occur very early. We have projects understanding what are the earliest events in colorectal cancer. That can be harnessing, the immune system to target evolution. That can be, combination therapy. Most likely this will be all these things together. Most importantly, we are trying to see whether we can, understand what RAS does in colorectal cancer. It's not known well. So for that, we have developed a lot of new tools that allows us to, understand what happens when we suppress RAS in colorectal tumor. So that's a very frequent oncogene and has not been drugged in colorectal cancer. We have also projects trying to understand the early steps of tumor progression and how we can use that to develop new therapies. And finally, we are thinking a way of using tumor heterogeneity against tumor. So evolution against evolution. But that's too preliminary to say.